hey, wow, you've been there this whole time? Why didn't you say something? Well, since you're here, we might as well get ready for our photosynthesis lab, okay? So, our photosynthesis lab is going to help us see how the effect of light, the effect that light has on carbon consumption in the process of photosynthesis. I was stuttering. So, as humans, we can easily get our food from things like this Lay's potato chips, right? But plants, they need to undergo the process of photosynthesis in order to get their energy. So, how are we going to go ahead and test the effect that light has on the consumption of carbon dioxide? That's a really great question. I'm going to go ahead, put on my goggles and my lab coat to stay safe, and I'm going to go ahead and show you how we're going to do that in this lab. So, in order to tell whether carbon dioxide is present, we're going to need to use an indicator. So, as you can see over here, I have this neat indicator, and it's bromothiamyl blue, okay? So if you look over here, it says BTB, bromothiamyl blue, and this indicator is going to help us determine whether carbon dioxide is present in the solution. So, as you can see over here, I'm going to show you the color of bromothiamyl blue before we put carbon dioxide into it and what it looks like after. So, I have a test tube, and I'm going to put a tiny bit of bromothiamyl blue into this test tube, and I'm also going to dilute it with some water. Okay, so as I'm diluting it with water, you'll notice that bromothiamyl blue has this nice blue tint. Okay, so it's got a nice blue tint. Can you see the blue tint? Okay, so bromothiamyl blue, once we dilute it with water, has a nice blue tint. Now, earlier I blew into this vial of bromothiamyl blue, this test tube of bromothiamyl blue, and I blew in carbon dioxide. Remember, as humans, we breathe in oxygen, we breathe out carbon dioxide. And as I put the carbon dioxide in, it changed from a blue color to a green color. Okay, I'm going to go ahead and do that here as well so you can see the difference. Okay, so just observe. So you saw that color change? You see how it's a dark green? If I keep going, it's going to change to a light yellow. Okay? So now that you've observed that color change using bromothiamyl blue, we're going to go ahead and make a bigger quantity of it in order to test it out in our test tubes. Okay? So, we're going to go ahead and use this bromothiamyl blue. We're going to put it in here. And I want you to see what I'm doing with this. So, I put about 100 milliliters of water in here. Okay? And I'm going to go ahead and put about 4 or 5 milliliters of bromothiamyl blue. So, let me set that up. Okay, so that's about 5. Okay, so I got 5 milliliters of bromothiamyl blue. I'm going to go ahead and mix it with my water. Okay, so I'm going to go ahead and stir it. Now I'm going to use this super sipper. Okay, super sipper. And I'm going to mix it up. And I'm going to breathe carbon dioxide into it. So we're going to load it with carbon dioxide. So again, notice the color change. Okay, can you see that? Notice the color change. Okay, so I'm going to breathe a little bit more, but this should be just about enough to help us get started. Now all we need is our plant. Okay, so here we got a nice little plant, and I'm calling this plant Atlantis since it's an aquatic plant. Okay, and Atlantis is going to help us because it's going to sacrifice a couple of its leaves for this experiment. So what we're going to go ahead and do is I've already placed a couple of Atlantis' leaves in these test tubes. And I'm going to go ahead and put this this mixture of bromothymol blue with carbon dioxide, and I want you guys to predict what you think is going to happen when we put it here in these test tubes, okay? Also, what we're going to be doing to differentiate and noticing the effect of light on these test tubes is we're going to put these test tubes in, two of them, these ones are going to be put underneath light, so direct light, and then these other two are going to be put in darkness, in that cabinet right there next to the dispenser. So, I'm going to put them in light and in darkness, and I'm going to fill them up with the bromothymol blue and carbon dioxide solution, and then we're going to notice what happens after a day. So again, with keeping our control variables the same, I'm going to go ahead and put about the same amount of mixture, of solution, into each of these vials. Okay, so this is our control one. That's our, this is our control one for the dark. And then the one for the dark with the Atlantis leaf. So, again, I have about the same, you can tell it's about the same. You can notice that the color is this nice green tint, okay? So I'm going to take some pictures, and like I said, I want you guys to go ahead and predict what's going to happen, and I'll submit that video, give it to you guys, and you guys have to submit the lab sheet as well. So, take care, look for the videos coming up, and I'll see you soon. Bye. Okay, so here is a picture of the Atlantis leaf in the cabinet along with our control tube. Okay, so this is the control inside the darkness, and over here we have the Atlantis leaf in the direct light. Okay, so direct light and the control as well. So go ahead and make some predictions. What do you think is going to happen? And then make sure to fill out your worksheet, and feel free to email me if you have any questions, and we'll discuss this in class tomorrow. Okay, so take care everyone. I'll see you soon.